This Week in Microbiology is brought to you by microbe.tv and is supported by the American Society for Microbiology. To learn more about microbes, visit microbe.tv and asm.org. This is TWiM, This Week in Microbiology, episode 334, recorded on June 12, 2025. Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from St. Louis, Missouri, Petra Levin.
It's great to be here.
Is it really warm there?
It's a little humid. It's not super warm. It's overcast, and I'm wondering if I made a mistake not bringing an umbrella. But slowly they are removing trees and stumps, and although we've lost a lot of trees, it's getting a little better in terms of overall appearance here since the tornado.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Well, hello, everyone. And it's overcast. It was raining earlier today, and it's now a little bit cloudy outside, and it's fairly pleasant, but the humidity came down in liquid form today.
Here it's 30 C. Wow,
Ooh.
And sunny.
It's summer.
What's the humidity? Where do I have to go to find that? 49%. I guess that's not bad, right?
That's comfortable.
That's great.
That's great. I don't know. I feel a little humid because it's not been this way for a while.
From a Missouri perspective, it's great.
But you're still wearing a turtleneck.
Exactly.
It's not even just a turtleneck. It's a thermal turtleneck, Michael. Because air conditioning everywhere is really cold, right?
Your office is no different than my office. I sort of have layers I go to work in, and people look at me like I'm crazy.
The incubator, you cannot regulate the temperature. It's whatever the building wants, right? So it's always cold. And then the trains are cold too. So I just wear this and I'm fine because I'm outside very little.
Mm-hmm.
It doesn't matter, but at some point I should stop. But it's because it's weird. I used to have a colleague at Columbia, Arj F. Stradiatis. He was a Greek scientist, very good scientist. And he always wore black pants and a black shirt and a black hat. And I always thought he was weird. Now I'm weird. If you like these programs where we talk about real science, no lies, no misinformation, no pseudoscience, we'd love your support to help continue them. We don't do ads. We have no source of income except your donations. And so if you go to microbe.tv/contribute, you can find out multiple ways where you can help us. The more of you, the better. Today we have, for your listening pleasure, of course, a snippet in a paper, and it's my honor to do a snippet about Brooklyn.
Brooklyn?
I always remember how many scientists, when they get up and they give a talk, they tell they're from Brooklyn. So many scientists came from Brooklyn.
I know, but the scientists in my family all came from the Bronx.
The Bronx? So, wow, that's
Bronx.
Unusual.
I know, exactly.
That's
so
unusual.
I feel a little fighty when people say Brooklyn.
Well, the paper today is in the Journal of Applied Microbiology. It's called Metagenomic Interrogation of Urban Superfund Site Reveals Antimicrobial Resistance Reservoir and Bioremediation Potential. And the first author is Sergios Orestes Kolotronis, and the lead author is Elizabeth Henaf. And these are individuals from Brooklyn, of course, as you will see, Weill Cornell, Rutgers, Asheville, North Carolina, the Fruit Studio, who knows what that is, Charlottesville, University of Virginia, and NYU. So this is all about the Gowanus Canal. The Gowanus Canal is about a 3-kilometer long canal in New York City. If you look at the map that's in the paper, you can see it starts inland and then goes out to the water, to the ocean. Not an ocean, but the bay out there. And it used to be a big transportation hub, right? It brought things to the interior and back, but it's being used less for that these days. There are small boats and tugboats and barges on it. But it is the most polluted body one of the most polluted bodies of water in the U.S. What a distinction, right? At the bottom is a thick layer of contaminated sediment, which is 150 years of industrial pollution and sewer overflow. This toxic sludge is called black mayonnaise.
Ooh.
It's full of coal tar, heavy metals, and industrial chemicals that were banned ages ago. They're still down there. So it's been declared a Superfund site, right? The Environmental Protection Agency identifies all these sites and puts money into them to try and clean up. And the cleanup for the Gowanus is about 1/3 complete. But they want to get rid of all this mayonnaise at the bottom, but there's still thousands of cubic yards. And of course, when it rains, untreated sewage gets dumped
More
into
comes
the
in.
canal. More comes in.
Right. So do people know, like a lot of in American cities, a lot of times when it rains, there's just overflow.
Yeah,
If it
they
rains a
don't.
lot, there's overflow from the sewers right into the waterways. Yep. This is a standard system. It's not.
No, I've seen it happening. I used to live in the city, and I would stand on the East River and see feces going out into the river. I'm like, and there's a guy fishing downstream from it,
Mm-hmm.
Oh,
well, you know, in South Carolina, we have signs posted at our rivers so that people who may have not grown up here know not to eat the fish out of the rivers because of the contamination. And especially when you think about eating fish that people eat, they're generally at the top of the food chain where they bioaccumulate many of these toxins. And consequently, the fish is actually harmful. It's not good for you to eat. And so, especially for individuals who are pregnant, and we warn them at every boat landing in South Carolina not to eat the fish from the rivers.
Right.
Man.
But I was always just amazed that, like, even now, like, I mean, cities all over the world, it's not an American thing, that this is the way to deal with too much rain that runs into the sewers is just to, at that point, have them go into the waterways.
That's right. So just to give you an idea what's in this sludge: arsenic, copper, lead, iron, polychlorinated biphenyls, chlorinated solvents and fungicide, carbon tetrachloride, ethylene dibromide, polycyclic or aromatic hydrocarbons, volatile organic compounds, salts, including brine and tartaric acid. The goal is to get rid of all this sludge at the bottom and cap it off. It's supposed to finish in mid-2025, but I don't think they're close to finishing. So the reason that they're studying this is because it's an extreme environment. It has both built and natural components, right? And this is a city, the most populated city in the U.S., so you can imagine what's going on there. So, and microorganisms always respond to challenges, right? When you have the Deepwater Horizon spill, all the microbial life readjusted, both in the water and on shore. And many of the microbes that repopulate can be used for bioremediation. So they wanted to study this site to see, first of all, what's the species diversity? This is a microbiological study. What's the potential for bioremediation, and can they discover new got all these biosynthetic gene clusters. In other words, sets of genes that can make something that could be useful, right? So they went into the Gowanus Canal. This was a number of years ago. They sampled the sediment. They do it in a canoe. They tell you it's done from a canoe. They have a PVC pipe that they drive 10 to 15 centimeters into the sediment. They put a cap on the top, and then they pull it up and expel the sediment into a 50 ml sterile tubes. So they do that for the surface, and they go down deeper as well. So they get the sediment on the surface and what they call the sediment core. And they took samples at 14 different locations along the whole canal at two different times, December 2014 and April 2015, both sediment and core samples. And then they extract DNA and do DNA sequencing. Then they, of course, analyzed the sequences for what's there. Species abundance. They identified 455 species across all the samples up and down the canal, including 13 archaeal species, 241 species of bacteria, and 201 viruses or bacteriophages, which I don't really talk about much. And 85 species are shared across 3 sample sets, and the 2 sediment samples share 128 species. And what they find is interesting, a lot of things, but one is that the samples from the core are different from the samples from the sediment, which makes sense, right? As you go down, it's less aerobic, I presume, right?
Oh, yeah.
So that would influence that.
Should be, very anaerobic by the bottom of that thing.
It's actually redox. It's all about redox because they want to throw their waste electrons away. And as the EH, or the electromotive potential, as you go deeper into the sediment, it gets harder and harder to throw away electrons, which is why we have methanogens and hydrogen evolvers, because you make a gas and it goes off.
So, by the way, this canal is brackish, right?
Mm-hmm.
It's because it's open to the sea, but then as you get up away from it, there's a little bit of fresh water up there from runoff and so forth. So it's a mixture. It's, brackish, as we call it. So the environment is a combination of the original sediment, 150 years of industrial waste, 150 years, and inputs from runoff, as well as the tidal influx, right? The tide rises and things go into the canal, and that contributes to what's in there as well. So they actually found marine microbial species, right? They list them, but I'm not going to mention them because I don't know any of them. Modestobacter marinus, Draconibacterium. I kind of like that. It's Draconium.
Mm-hmm. They
and
have
they also
very
find
good names.
freshwater sediment species like methanobacteria and micrococcus. And they also find soil microbes, micrococcus and others. So I guess they're washed in from the soil, right? They also find extremophiles, right? They find psychrophilic, right?
Things that like to grow in the cold.
They find thermophilic, things that like to grow at high temperatures, right? And halophilic organisms, things that like high salt. And this canal is determined to be high salt, hypersaline environment. And some of these organisms that they find, they list the psychrophiles, the halophiles, and the thermophiles. Some of these have bioremediation properties. So again, they're probably there because the bacteria are responding to this environment. And maybe you could get them and use them for other things as well. Now, as we've already said, untreated wastewater is released into the canal. I mean, right? Untreated human waste. So what do you think they find?
Pathogens.
Human gut bacteria.
Gut bacteria.
Gram negatives. Acinetobacter. Oh my gosh.
Oh, yeah. Well, it likes to eat plastic, so there may be plastic in there too.
Gram positives. Clostridium. Let's see any of the Gardnerella. Mycobacteria. Streptococci. Lactobacillus. Amazing. And of course, it's both archaeal and bacterial isolates. How about antimicrobial resistance? You think there'd be any antimicrobial resistance genes in there?
Absolutely.
So they identified 28 genes within 8 classes of antimicrobial resistance. The most common ones are rifamycin, also aminoglycosides, tetracyclines, alphamycins, phenol sulfonamides, and multi-drug resistance genes.
Well, the RIF resistance is what actually surprised me, because rifampin is not a drug that physicians indiscriminately prescribe. You have to really want to prescribe rifamycin. It's reserved for serious infections. It's reserved for tuberculosis. And you really have to want to do it. Plus, it has all sorts of off-target effects in the patient, so they don't like taking it. So I was actually surprised that they pulled that cluster out, and it got me to thinking, what's going on with the RIF resistance? What's keeping that present in this community of microbes that can degrade anything, including, you haven't gotten to this part yet, but the heavy metals?
So, right, exactly. That's the key, is the heavy metals. So is RIF resistance, and this I don't know because we don't work with RIF or rifampin in my lab that much in the resistance. Is the resistance a genetic mutation in its target, or is it a cassette, like a drug resistance?
That I don't know off the top of my head.
I feel like it's mostly a mutation in its target.
Yeah, they say it's a single gene.
Yeah, and so that also makes it very interesting because a lot of them, and they'll get to the heavy metals, right? These sometimes are co-regulated or come in on the heavy metal resistance and some other drug resistance cassettes.
Yeah, they come as a
RIF
cassette.
Doesn't, I think. Yeah, this TET resistance in their figure, I think it's supplemental figure 3, is their biggest. It's really scary, especially since that drug also not used really for the past 15, 20 years as very much.
Shouldn't go swimming in the Gowanus Canal.
Really, any canal that runs through a city, unless they've really cleared you, I'd probably avoid.
Yeah, we tell that to RFK Jr., right?
Yeah. But I know people have gotten in London. People go swimming in the Thames, and or the people got sick in the boat race on the Thames. And again, I think it's this, and they talk about the sewage overflow problem. So, but again, I'm curious about the RIF.
So next, they look at bioremediation metabolism. They look at the genes and they put them into metabolic pathways as defined by databases. And they can find 64 pathways that are related to the canal's history of contamination. And aromatic degradation is the most represented, with 52 pathways, including compounds like toluene cresols, catechol, and phenolic compounds. Oh my gosh, that was such toxic stuff.
Well, they were preserving wood.
Yes.
They were making telephone poles.
That's right. Okay, so what kinds of, so within the aromatic degradation class, which we said was the most represented, toluene degradation is the most relevant subclass. What do you use toluene for, Michael?
Metals. You use it for metal cutting. It's used to effectively tool and die. It's the cutting fluid.
And also,
or the solvent is the part of the cutting fluid. It's not specifically toluene. It's in the class of the volatile organics.
They also have, so these are gene clusters that degrade these compounds, right? So toluene, catechol, phenolic compounds, and they can associate these with specific microbial species in many cases. So those are the aromatic compounds. They also find pathways related to degradation of C1 compounds, alcohol, formaldehyde, and others.
But nothing to go after cement shoes.
No. And there's a section called Heavy Metal Superstar.
Oh, yes. That one intrigued me. And, well, before you get to it, this one is why we do science. If you think about what the country has been talking about over the last year, 2-3 years, it's all about rare earth metals. And if you have a microbe that can mine metals and deal with heavy metals, like these compounds that Vincent's going to introduce us to, like cadmium chromate and lead and mercury, and pull out the rare earths so you can effectively purify it. And you can actually do some useful work from this sludge.
Yep. So again, they're looking for genes that, or clusters of genes that metabolize these heavy metals. And so they find 3,153 genes encoding proteins associated with heavy metals. The largest one is iron. I guess that's not surprising, right?
No. New York was big into tool and die making.
Now it's all digital, right?
Or 3D printed.
So iron is the biggest. Then we have cobalt, copper, nickel, cadmium, chromate, lead, mercury, vanadium, arsenic, and aluminum. Would have been nice if they talked about what these were used for in the city, right?
Well,
like you just did.
Stainless steel is a big one. You have the chromium, you have the vanadium, you have cobalt, and that's effectively how you make stainless steel. They're it's an alloy, and so in fact, stainless steel was a creature of the United States. It came out of here.
Yes, and then other countries underpriced us.
That's unfortunate but true. It's hard to keep a secret when they're fundamental principles of science.
Of course.
Because if you do science, you can figure stuff out.
But even if you can't figure it out, you can always bribe someone.
I think when you have this many plants making it at some point during the 19th century, other people are going to get that tech.
Anyway, these metal-associated genes, which they call them, they can associate with about 200 unique bacteria, like disulfobacteria, methanobacterium, etc. Okay, and then finally, they want to know if you could identify gene clusters that could make something that's useful, BGCs, and they can do that. There are databases that they can query, and they divide the compounds that could be made from the genes that they find into 6 classes, depending on their origin, like polyketides, non-ribosomal peptides, ribosomal synthesized peptides, terpenoids, saccharides, indole alkaloids, etc. So they identified 2,300 gene clusters that could make something. Sometimes they're known compounds that they can make, and they say 1,600 of the gene clusters were not associated with a specific compound, but might encode potentially unknown novel compounds that could be useful. So someone can take these data now and mine it and try and do that. So this is basically the paper. You get a nice understanding of what's in there in terms of microbiology, but also bioremediation. Maybe that's useful for another site. They say you could take these species, grow them up, and use them elsewhere, and potential molecules that you could make. So they say the Gowanus is a reservoir for microbial mechanisms potentially important for biotechnological applications.
I mean, what I saw this as actually was like the Gowanus, at least in its current state, which hopefully won't be for so much longer, but that it is essentially a selection. It's been doing selection for some bioremediation bugs that people have been looking for a long time, but here they are. Right here, you could probably pull them out and do a couple more rounds of selection and get ones that do a lot of different metal transformations. It'd be pretty cool.
So they end the paper, which I thought was a very interesting sentence. They're talking about growing up bacteria and putting them into other sites for bioremediation, and they say this approach requires considering the ethics of interventions on non-human organisms, because even though we can't relate to microbes in the way we can relate to beasts of burden, harnessing them still implies making resources through labor. Oh my gosh, I've never heard anyone talk about that. Bacteria, putting bacteria to labor? I mean, is that a bad thing?
We've been doing it since the beginning of time. So the next time you open a bottle of beer or eat a slice of bread, think of the microbes that you have exploited.
I mean, I can understand what they say, beasts of burden, right? Or even lab mice, right?
Are they going to start carrying teeny tiny little protest signs?
I thought that was very interesting.
Well, I was reminded when I read that sentence of my old PhD advisor when they announced the extinction of variola, the agent that causes smallpox. He says, "Where were the protesters to protest extinction of variola? Where were the protesters? We eliminated."
Well, that's true. By the logic in that sentence, if you eliminate a species, it's not good. You have to consider the ethics of it, right?
And the ethics was smallpox killed billions of people over the millennia.
Yes. Although I think in the case of microbes, there's always another microbe to fill its niche. I mean, I feel less, although I have special microbes that are special to me, I do feel like less sensitive about whether they're eliminated or not. So maybe that's wrong. I don't know.
It's an interesting approach. Okay, so now You've heard this in the news. Michael's going to tell you all about it.
Well, this is today's paper is going to address a topic that we haven't covered much on TWiM. And my appreciation of the need to cover this topic arose from a story I heard recently on the evening news, where the soundbite that the anchor teased the audience with was that a Chinese couple traveling through the Detroit airport was charged with smuggling a biological pathogen into the United States. No, it wasn't a new variant of SARS-CoV-2, but rather it was a plant pathogen, specifically particularly nasty fungus, Fusarium graminearum. When I heard it was that particular fungus, I was transported back 23 years into the past when I was on the state of South Carolina's governor's task force on biodefense issues. This was shortly after the anthrax attacks of 2001. And this is one of those microbes that kept me up at night after one of our meetings. Why? Well, I learned at the time that Fusarium graminearum is the primary cause of Fusarium head blight, an extremely destructive disease globally, affecting major cereal crops like wheat, barley, maize, and for the city boy, that's corn, and rice. And this disease leads continuously to substantial yield losses and significantly diminished grain quality. The U.S. Department of Agriculture estimates it alone, it costs over $1 billion in losses to U.S. farmers in the U.S. to their wheat and barley crops. A widespread outbreak of this microbe on those crops could severely impact any nation's food supply and economy. If that were enough of a threat, appreciate that this fungus also produces not one, but 3 mycotoxins, which is a fancy word for a toxin made by a fungus. The first one that this creature produces is a particularly nasty one, deoxynivalenol, which is also referred to as DON. And toxin DON is also known as vomit toxin. So you immediately know the consequence of ingesting grain that has this toxin. The other 2 toxins it produces, nivalenol and zearalenone, can be both toxic to humans as well as livestock, where again, upon ingestion, the symptoms of vomiting, diarrhea, abdominal pain, headache, fever, liver damage, and in some cases, reproductive defects if you're exposed chronically. And this then also, chronic exposure can lead to impacts to the host immune system of the infected individual or animal. And if that weren't enough, there's also linkage to teratogenesis, which will lead to birth defects in your offspring, and of course, cancer. Now you know why it kept me up at night. And as you might imagine, if a nefarious group grew up just a little bit of this fungus, they could easily cripple an agricultural system of any community that they happen to release it. And a covert attack could go unnoticed for weeks, resulting in the spread of the fungus and a potential for an epidemic against our crops. I want to say up front that beyond the news story, I have not learned any more of the details beyond the arrest of the individuals mentioned in the news story for allegedly smuggling Fusarium graminearum into the U.S. and what they were going to do with the particular strains they brought in once the fungus was here. But the smuggling occurred last July 2024, with one of the individuals being immediately sent back to their country of origin and the other arrested only just last week. Hence, it made the news and gave me a new nightmare. I'll leave the courts to sort out their intent and whether or not a crime was committed, but I thought you, our listeners, would like to know first how we, the community of science, ensures that the work we conduct in our laboratories is done in a safe and, most importantly, ethical manner, while at the same time exploring this topic of fungal plant pathogens and how we, again, the community of scientists concerned with ensuring that we'll be able to continue to feed ourselves, are addressing this. And for us here at TWiM, this is albeit an orphan topic, and so we're going to try to keep you up to date as to what's going on in the microbial world. In the United States, universities self-police through their institutional biosafety committees that all must follow a minimum set of guidelines that were additionally laid down at an Asilomar conference in the 1970s. And then they were subsequently discussed and went to public hearings, and they were ultimately codified by the National Institutes of Health as the biosafety rules and regulations, primarily which were initially developed to address the brave new world of recombinant DNA technology, and that resulted in the NIH establishing guidelines for research involving recombinant or synthetic nucleic acid molecules, which outlines the practices for handling recombinant DNA and synthetic nucleic acids. These guidelines are, let me underscore this word, mandatory for any institution or any individual receiving NIH funding for this type of research. The guidelines also address biosafety levels, which are a tiered system for containment practices based on the risk associated with the biological agents, which again greatly assuage my nightmares. And at the time, I was biosafety chair of our institution for over 17 years. So I saw everyone's application to the biosafety committee. And I can say with certainty that my colleagues were practicing safe science. They were always rigorous about submitting materials to the committee for review. And most importantly, we had 2 members from the general public who were trained to recognize when things weren't right, in addition to having university faculty on the committee. So this was a committee that effectively was concerned with you, the public, ensuring your safety. And I'm going to drop into the show notes some guidebooks. There are 4 biosafety levels that define proper laboratory techniques, the safety equipment that we all must use, the design of the laboratories that we have, and they all depend on the types of agents being studied, and they are laid out in this comprehensive manual. It's a free download for everyone. It's entitled The 6th Edition of Biosafety in Microbiological and Biomedical Laboratories, and it's available from the CDC, and it's still up at the CDC site. And again, it's in the show notes.
Right. And I think it's important to note that even though the universities also are supposed to police this, excuse me, when you go through NIH review, you also have to have essentially approval from your university. So there's a second level of review. And then there's a third level of review, which is also important. People know that there's certain organisms, ones that have to do with potentially being associated with warfare, bioterrorism, that are called select agents. And those are very special because those have to be checked when you actually review an NIH grant, if there's one of those in there. So it's not only the university policing, the NIH also has a level of review because that's one of the questions that you're asked when you review. Is there biosafety problems? Is there proper things in place? And then the select agents. So every level. Yeah. So just to make sure it's not just the universities.
Mm-hmm.
And these aren't silly little rules. These rules have teeth. If you violate the select agent rule, the university is fined $250,000 for each violation, and you, the scientist, are personally liable for $250,000 for each violation. So it's, as they say, serious as a heart attack, folks. We worry about the safety of the general public. We take it all very seriously. And there, as Petra was saying, there are 4 risk groups, and they follow along, and you can go in to see what's actually going on when you review the biosafety site. But I should point out, for animals and plants and other multicellular organisms, like Fusarium is going to attack a plant and not a vertebrate animal. And so there are different guidance for that, and that's managed by the USDA. and not necessarily the NIH. And they similarly have select agents that still come with the same set of penalties. And when I was reviewing the USDA website this morning, I found another paper we need to cover on TWiM. It's from 2 years ago, and its title on the USDA site was "The Virus Undercuts Fungus Attacks on Wheat." And here a mycovirus, so this is not a bacteriophage, but a phage against a fungus. It disables the fungus's vomitoxin making machinery. And so I dropped a link to that particular story into the show notes. But as you likely surmised, we're going to cover one particular paper today, and it's entitled "The Mechanisms of Developing Fungicide Resistance in Fusarium graminearum, Causing Fusarium Head Blight and Fungicide Resistance Management." It's by Melania Jawananda and Delanthea Fernando, and they're at the Department of Plant Science at the University of Manitoba in Winnipeg, Canada. And it's a really neat review that they offered to us. And it helped me appreciate that my nightmare is actually being aggressively addressed by a body of scientists. I found a lot of literature around Fusarium because it's such a significant agricultural problem. There's a body of literature out there developing around Fusarium and how best to control and limit its impact on its ability to grow these nutritious and toxic-free grains. And again, I want to point out that this isn't some just minor annoyance, a cosmetic thing. It's a major economic threat to small grains like wheat, barley, oats, and corn, and it's a global phenomenon. And in a 2025 pathogens paper, a group has begun to investigate how this pathogen is impacting wheat management in Central Europe, if you will, the former client states of the former Soviet Union, because we didn't have much data on what Fusarium was doing in those former Soviet client states. The paper I selected is a good place to start because it assuages first my fears and hopefully yours, and it helps us to begin to think about the sort of science that needs to be conducted to ensure that we'll be able to continue to feed ourselves. The review starts out with a familiar story that should not come as a surprise, that the overuse of an antimicrobial leads to the emergence of resistance by the pathogen that the intervening agent is attempting to kill. This is a familiar story to listeners of TWiM, as antibiotic resistance is significant, if not the most significant issue facing the infectious disease community that treat people. It's no different in the ag world. In the paper from Pathogens that was published in late 2024, Jawananda and Fernando offer a compelling narrative of how this fungus is fighting back against the fungicides that the ag industry has developed to help and that we use to protect our cereal crops. Again, fungicides are meant to kill fungi, responsible for something called Fusarium head blight. And I also dropped in the show notes what it actually looks like when you have infected grain. The term that the caption used is the grain or the seeds of the wheat actually look like tombstones. And they really look like tombstones that you would see in a graveyard standing up. I should also mention that resistance to antifungals is not unique to Fusarium resistance. It has emerged amongst human fungal pathogens, with Candida aureus now responsible again for giving me nightmares, as it's such a challenging fungus to inactivate once it establishes a beachhead in the built clinical environment. And I've dropped a slide deck from the CDC on that problem into the show notes. So now back to Fusarium head blight. The cornerstone of Fusarium head blight management for a long time has been the use of fungicides, with them being quite effective at cutting down both the head blight as well as contamination of the healthy grains with those nasty mycotoxins. But as the German philosopher Nietzsche offered, what doesn't kill you makes you stronger is certainly at work here in this situation where the pathogens of the fungi are becoming increasingly resistant to the common fungicides being used to manage FHB or Fusarium head blight. Why? Simply, it's through this immense selection pressure that the fungicide is placing on the fungus. Live or die is the question the fungicide is asking the fungus. And Fusarium germanium's population is effectively responding. And we have seen this only increase with the advent of these mega farms of large scale acreage being solely planted with one grain type or one strain type of grain. And we have witnessed the inevitable, the development of fungicide resistance. It's a classic evolutionary arms race. It's no different than the evolution of resistance that we have seen when we overprescribe one particular antibiotic in our clinics. Fortunately, there are many commercially available fungicides targeting this fungus. And like the highly active antiretroviral therapy for HIV, each fungicide targets a different biochemical process within the fungus. And this is all coordinated by the International Fungicide Resistant Action Committee. And I love their acronym. I normally hate acronyms, but this one is referred to as the FRAC Committee. And the FRAC Committee has grouped the fungicides by focusing on the mode of action and resistance risks. I like the next sentence from the review, for it helped me really get a good night's sleep after that story broke, as their explanation enabled me to appreciate the best targets to think about as the authors walked us through that all hope is not lost, while at the same time allowing me to draw parallels to my knowledge base of how the scientific community continues a targeted approach to developing antimicrobials to combat multi-drug resistance bacteria in the clinic. The sentence, according to FRAC, there are several modes of action based on nucleic acid metabolism, cytoskeleton synthesis, motor protein, respiration, amino acid synthesis, protein synthesis, signal transduction, lipid synthesis, cell wall biosynthesis, cell wall melanin synthesis, host plant defense induction, unknown modes of action, chemicals with multi-site activity, and biologicals, the virus, with multiple modes of action. So in their first table, they offer us up 6 common fungicides used to control FHB and its target fungus. And they list them off, and you can see their mode of action and what they're specifically targeted. One particular fungicide, a demethylase inhibitor, which are the trizoles, the imidazoles, they go after sterol biosynthesis, which is going to effectively go after the membrane, and it specifically goes after cytochrome. You have the quinone outside inhibitors. They go after respiration because the fungicide inhibits the cytochrome C respiration system. And they list the other 4, but I think you get the idea. There's a different target. And so if it becomes resistant to one, we still have others. Or like HIV, if the mutation rate is especially high, we can go to using more than one fungicide. But again, in farming, it's all about the cost of farm to table. So unlike HIV, where it's the cost of a human life, so we're willing to spend the extra cash, in farming, they may not necessarily be willing to go to the highly, the multi-drug approach, but we may have to. The bulk of the review then offers us this great tutorial on how the fungicides are becoming tolerated by Fusarium. And this fungus has quite a few tricks up its sleeve. And these tricks are not any different than you already probably know. Think about it. Target site alteration. This is the most common way of having resistance to a fungicide in the fungi. It simply is the alteration of the fungicide target. This alteration is achieved in the fungal genome by simply selecting mutations in the target site during DNA replication. These mutations change the amino acid sequences that result in an altered shape of the target site. And they give us a great figure to help us think about these things. A metaphor that is often used to explain target site alteration is the fungus mutates its fungicidal target. It's effectively rekeying the lock so that the fungicide can't get in. And the fungicide is the chemical key. So if you rekey the lock, the key no longer will work. So it can no longer inactivate the fungus because the inactivating agent is unable to interact with the microbe. This is the common approach, one approach of resistance emergence that we have seen time and time again in HIV. We see it in SARS. That's what all the variants are about, where they're changing the spike protein. They're just changing their spots. It's a simple amino acid change that can lead to resistance. The one that comes to my mind in the bacterial world took a long time to develop, and that's because of the number of genes that had to have compensatory mutations. And that, of course, is the resistance to vancomycin, where resistance to this drug results from the alteration of the entire peptidoglycan synthesis pathway in resistant bacteria, where the terminal amino acid residues of the disaccharide, the N-acetylglucosamine, N-acetyl-muramic acid in the side chain, that typical D-ala side chain that's hanging off, changes to a D-alanine D-lactate. And this reduces the vancomycin's ability to bind to this peptidoglycan precursor that will then prevent it from extending the size of the peptidoglycan, and you inhibit cell wall synthesis. So this is what these fungi can do against some of the fungicides used today. We also have target protein overexpression. Again, imagine, using the same metaphor, if the fungus just built a whole lot of more of the target protein. Again, metaphorically, is you duplicate the lock, so now it's a concentration thing. The key has to try all the locks to see which one will let the fungicide in to kill the fungus. Even if some are hit by the fungicide, there are plenty of other fungi that have not yet been killed that can, again, be selected to live through the onslaught of the fungicide application. We also then have detoxification. And the fungus, and this is what Vincent's paper was really talking about, is the microbes are breaking down these organic compounds into non-toxic forms. And in fact, this is the job of every fungus, degradation. So it's no surprise that they can degrade the chemicals that we make. And our strategy to making better fungicides or any pesticide has been to simply to decorate the molecules with halogens. Put a fluorine here, a bromine here, a chlorine there. By making it more electronegative, the dehydrogenases and hydrogenases have a tough time getting in. But fungi can even do more. They can make molecular oxygen grenades like phenarachidea, and they just throw out oxygen ions and explode and tear the chemical apart. That's how phenarachidea degrades lignin. So the fungi have all of these tricks, but they still have one more. They got an efflux pump. And finally, the efflux pump is nothing more than the microbial bouncer. The cells can actively pump out the fungicide as fast as it comes in before it can do any damage to the fungi. Here again, we're reminded that, well, that's how tetracycline resistance often manifests. So you might be wondering, why not just go organic? Well, selection pressure is really quite strict, and these fungi just seem to get around, and it's really next to impossible to go after it. The authors emphasize we need some serious mitigation strategies to delay resistance. And we have already seen some of these strategies applied in the clinic. They're called antimicrobial stewardship approaches. And this is indeed what the USDA is doing. This is indeed what many of the papers in the literature are talking about. And that is, of course, regular monitoring. They're keeping a close eye on fungal populations to detect sensitivity shifts so you can swap out the fungicides to one to which the fungus is sensitive. We're arguing for responsible application. This means following manufacturer recommendations, using minimal sprays, and importantly, rotating and mixing fungicides with different modes of action. Now we have the promise of AI. So we can do disease forecasting models using predictive models based on weather and wind and crop phenology. This can help growers decide when and how much fungicide is truly necessary to prevent from applying too much selection to the fungi that we're trying to get rid of. Which gets me to thinking, will AI help us in deciding how best to deploy a combination of antimicrobials that may have different targets to better address resistance emergence? And this is where a paper like Vincent's discussed really comes to play, because again, we are not just dealing in a vacuum where it's just Fusarium. There are other pests attacking our crops, and often the farmer or the ag business is using an integrated pest management, which is a holistic approach combining host resistance and cultural practices like crop rotation, and of course, the judicial application of these antifungals. And this is what is essential for long-term stability. Ultimately, this review serves nothing more than a critical reminder that we're in this constant tug of war with these microbial masters of adaptation. There are more of them, and they divide faster than we can think, and most importantly, feel the solution, especially given that they are facing the tremendous selection pressure of live or die. And understanding their resistance mechanisms and implementing smart, integrated strategies are going to be our best bets to keeping our crops healthy and our food supply secure for many years to come. So that's it in a nutshell.
So probably the strains in China are different from the US, so it would be not a good thing to bring them here, right?
That is correct.
Yeah, I feel like this is the same reason we're not supposed to even bring like an orange. You know, you have to be careful when you grab something for breakfast if you're traveling transatlantic or transpacific. I mean, this
or
is
even
like,
to Hawaii.
Or even to Hawaii, Australia. I mean, if you fly to a lot of places, Hawaii or Asia, they have careful controls for you personally, and we have mostly dogs. Actually, I think that they use. I don't know how they found these on these individuals, but it does seem from their text that they went through quite some ways to get around the system. But it is really scary, and I think we have mentioned before bananas being one of the crops that is essentially a monoculture. There's only one strain of bananas we all eat. And I think it's very difficult with that one because they're propagated from plant to plant. They're not, you can't even take them through a seed. So,
this is quite serious, and, you know, it's enough cause of concern that, again, we need to practice ethical and appropriate science if we're going to continue to have the public's trust.
Yes.
Yeah, a story like that doesn't help, right?
No.
Although I think most likely it's inadvertent, right?
This one doesn't seem, reading that article that you guys got posted, it seems like they were trying to essentially smuggle it in. I mean, probably for scientific purposes, but just don't do that. And especially since it's an agricultural organism that can do quite a bit of damage. But I mean, in a way, it is good because they did essentially get caught at the airport, from what I can tell.
At the Detroit airport.
At the Detroit airport, and that's what's supposed to happen. And I'm impressed that happened because, again, the dogs are sniffing out really fruit and meat and things that you're not supposed to bring in, and you're not supposed to bring them in for a reason, and that reason is a good one. So in this case, I think it feels like the system worked, which is good for us and good for our grain crops. So
in the old days, I always used to get asked, where did you go? I went to a virus meeting. Did you bring any samples back? Of course not.
Not intentionally.
But now that I have the global entry, you just zip through because you're a trusted person.
Yeah, but I think the dogs are still supposed to.
The dogs still sniff your luggage before you get out of the controlled area.
I know. I'm such a dog person. I always want to say, "Oh, you're so cute," but you can't do that because they're doing their job, and you don't want them sitting down because once they sit down, that means that they sniff.
That means you go into the room.
Yes, the room, and then they start going through your stuff. So, in any case, I think in this case, the system worked to not transport things. I mean, we had somebody transporting embryos. Again, that's a lot less risky, and probably should have been more of a slap on the wrist at Harvard. But this is really serious because it can do a lot of damage to our food supply.
All right. Thank you, Michael. All right. That's episode 334 of TWiM. You can find the show notes at microbe.tv/TWiM. If you like these programs, consider supporting us. Microbe.tv/contribute. Michael Schmitz at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Petra Levins at Washington University. Thanks, Petra.
Thank you.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.